Okay, then I'd like to ask Andrea if he can do whatever is needed to set up. Again, very pleased that Andrea is able to join us today. He's a group leader at the Institute for Molecular Medicine in Finland and an instructor at the Harvard Medical School and the Massachusetts General Hospital. So his research interests lie on the intersection between epidemiology, genetics, and statistics. And today he's going to talk to us about COVID-19 Host Genetics Initiative, how to enable data sharing across 120 studies and 600 researchers. Andrea, the screen is yours. Thank you.
Okay, thank you for inviting me. It's a pretty new topic for me, digital humanism. So I'm very excited to be here. I'm gonna tell you a little bit about our initiative, but since I thought that here would be more interesting for you to hear how we use informatics tool to enable this initiative and how I think this abundance of new tools of communication has allowed scientists to come together in a kind of unprecedented way. So what we mean for host genetics, just to clarify those that are not used to the terminology, we are really interested to study human DNA. And with viral infection, there is the virus genome and the human genome. And so we are on the side of the human genome, and we try to understand why some people develop severe COVID-19, why some people have less severe COVID-19, and how the genetics of the individuals can influence the severity of the disease or the susceptibility to COVID-19. Now, this initiative really started with a tweet on the 16th of March, 2020. At that time, we were just 3 groups that were interested in coming together and share genetic data to study the host genetics for COVID-19. And we really didn't know what will happen when we did that. And I think this is really highlight the power of social media. What we did to motivate people to kind of come together is that we decided to offer free genotyping, so free DNA sequencing on individuals that, or a researcher that will share a sample with the wider community. So when we started this initiative, we also wanted to set up some aims on what was the goal. The important things is that we wanted to be a bottom-up collaborative effort. I think in the human genetic community, we are really used to collaborate, and we have seen over and over that top approach imposed by grant agency or large organization tend not to work very well. And so I think bottom-up initiative tend to be a better approach. And here in the aim, you can see that our goal is not to directly to publish paper. Our goal as a consortium here is to share and provide resources for sharing. And so you see the first aim is to foster an environment of sharing resources, which was very important in the beginning of the epidemic, where many studies need to set up prospective collection and they didn't know which protocols to use, which questionnaire to use, and so on. Is to organize analytical activity, so support centralized as well as decentralized studies focusing on the host genetic. And finally, use a platform to share results of such activity to benefit the broader scientific community. The second building block of the initiative is certain principle of collaboration. And this is something we decided in the beginning to motivate people joining. And I go through them quite quickly, but I think, the collaboration in an environment of honesty, fairness, and trust. This was very important for us to promote early career researcher, and so many of the leading of the working groups are actually relatively young in their academic career. And I jump on 7 that we really not try to inhibit any work being done by individual studies. So we are not a monolithic initiative where everyone needs to adhere to certain standards, but everyone is able to do something and connect within the initiative. And so we also foster this type of collaboration. And I think seeking permission for other group to use result prior to public release is also one of the code of conduct. The other thing is that we wanted to set up a system where study could reach out and be registered, but a lot of these genetic consortia are done by just sending an email to your friends or your colleagues and trying to form a group. Here was really done through a more systematic approach. And we set up a registration form where we also wanted the study to really specify what were the research questions, so that in this way other study could contact these studies and start project and collaborations. So we have, if you go on the website, which it's up here, you see covid19hd.org. We did a browser basically where you can see all the registered studies. And what you can do, the first things to notice is that we have registered studies, so those that register and they're interested in joining, and then we have data contributors, so those studies that have actually been sharing data or summary statistics. And we highlighted them on the map. You see we have around under 60 study register, and then you can even contact directly the study through this button. And for example, Redict, which is running, is the principal investigator of the Estonian Biobank. You can email him, and if you see that there's something that you're very interested in collaborating, you can do that. Now, everyone, and this is something relatively new, at least for me, everyone in this initiative was invited to Slack. So we have a Slack channel. And that's a very, interesting way to communicate. I think when you have more than 950 people on a Slack channel, it's not easy to communicate. And so it became almost like a posting board. So slowly we've been moving to motivate people to create channels where they can spin off a research project that are specific to certain aspect of human host genetics. So flu-like symptom, you can see sex differences, pediatrics related, HLA, which is a region of the genome related to immunity, ancestry, and so on. So where are we now? We have around 928 members. There are around 300 members which are weekly actively on Slack, and there are around 170 messages sent per day on Slack. The website has around 41, I think around 44 now, unique user for more than 137 page view. And the contact button seems to be used quite well to contact the different studies. As you can see from the map of the study, we cover more than 40 countries, which is really pleasant to see that it's not only US and Europe, but we see from traditionally underrepresented country in human genetics. In human genetics, we have a big problem, which is they've been studying, in a sense, too much individuals of European descent, and we have not done too many studies on other population, which decrease the diversity of our studies. And so this I think highlight our other traditionally underrepresented countries also been spinning off some studies. Now, in terms of impact, I think it's been a good impact. NIH, which is the National Institute of Health, which is the largest funder of biomedical research in US, in this notice of special interest, clearly highlighted that the data produced in this specific call need to be shared and related to our initiative, and so this is a nice recognition. And finally, this was like was picked up by the New York Times, so we had a spike here in the number of visitors on the website. Now, we have two ways to share the data. Here is speaking about sensitive data, clearly clinical data and genetics data. There are two ways to share the data. We really want to motivate option one, which is individual share individual level data. This is done by using from the clinical data using a data dictionary, which we've been creating. and then depositing the data on existing repository for data. One is the European Genome Phenome Archive, which is hosted at EBI, MBL, and the other is the NIH AVIL. This is more for the US-based studies. The study PI remain the controller of the data access, but theoretically every bona fide researcher with a good research question can get access to this data. And so this is really important for us. And internally, we do data harmonization, quality control, and central analysis, and we make summary statistics publicly available. Now, this is something that is starting right now. Most of the studies still use option 2, which is sharing of summary statistics. It means that each study run the analysis and then share the aggregated, in a sense, data rather than individual level data. And we do a centralized meta-analysis. So we combine this aggregated data, and then we make these results publicly available. These avoid data to be transferred on the central repositories, as the disadvantage that other researchers have a hard time to access this data. This is more or less how it works. We have working group defining phenotype that we are interested in. For example, severity of COVID-19 or susceptibility of COVID-19, and then we have an analysis plan. These are posted on the Slack channel where we get feedback from the larger group. Once these phenotype and analysis plan are defined, they are combined with the contributor or summary statistic or individual level data. And then the analysis team crunch these data. And every few weeks, we post the result immediately on the website. I think this is essential. This is a big change compared to previous effort. We're not publishing papers. We share immediately the result and let others use this data to publish paper. The analysis is done currently on Google Cloud, since it's perform quite well and has a good security. So we have two types of centralized activities. We have two types of analytical activities, those that are centralized, which is a limited amount of analysis, which can really benefit from the maximum sample size of combining all this study together. And then there are the additional analysis that require specialized approaches or focus on specific patient groups. And these are spin-off by other working groups that are related with the COVID-19 Osteogenetic Initiative, but they can easily move out from it if they want. For example, Jean-Laurent Casanova Rockefeller, they're looking at monogenic inborn errors of immunity, and they have their own consortium. And so we sponsor all these type of side activities. This is an example of the phenotypes, not going too much in details. Just to give you an example, we are looking at hospitalized individual with COVID-19 versus non-hospitalized. It's relatively simple, phenotype definition, because clearly they need to be robust across many studies which have collected data or through the clinic or through electronic health records and so on. These are the result of the first meta-analysis, which we posted. So you can go on the website and download the results. From this meta-analysis, there were 977 COVID-19 positive cases and 633,000 controls, which cannot be 100% sure that are COVID negative. So some of will actually have COVID, but this is a relatively rough analysis. And if you're not familiar with this plot, each dot is a genetic variant on the genome. And this is a p-value, so higher you're up here means more significant. And we normally want the p-value to be very low, 10 to the power -8. So we are like up here. And we test basically around 15 million genetic variants on the genome. And we compare basically those that have COVID-19 versus those that do not have COVID-19. And you can see here it's each dot. There's no dot that goes above this line, meaning that we don't have any significant yet. So what we say here is that it's still underpowered and we will need larger sample sizes. Now we have creating browsers, so you can navigate the results directly. So look, for example, on specific mutation in certain genes. People are interested in ACE2 and other specific gene. You can click on those. You can see the type of mutation. If it's a missense loss of function, you can see the gene on the gene region. and the association. So this called locus zoom plot indicates higher again is the dot here, more strong is the association with the disease. We also, apart from producing scientific results, we produce resources that are available to everyone. We have made available questionnaires used by the different studies. I think we have more than 15 questionnaire or assessment tools that have been used by different studies. We have consent and protocol used by different studies. This is extremely important for studies that are setting up for the first time. They want to know what are the study have done, how they frame their consent form and their protocols. This is extremely important. We have data dictionary, and clearly we have a catalog of study that can be used in many different ways. And then we have these summary statistics. Now we're partnering up with several academic and industrial partners, which has been essential for I think, allowing studies that do not have financial resources to do this sequencing or genotyping of their sample. So at Erasmus MC in the Netherlands, as well as FIMM, we do free genotyping with the agreement that the researcher need to want their data widely shared with the scientific community and so deposit on EGA. Other pharmaceutical companies, biotech, and as well as non-profit organization, they also provide discounted sequencing for countries that really need it. We tend to favor countries that don't have economic means. So, for example, from Africa or South America, rather than supporting study from Luxembourg. But that's a decision we take. A FIMM, Erasmus MC, for example, accept almost every study. You can read more here about all these things. This is really a huge effort of many people. What I want to highlight is that there's been a tremendous support by people that through this community were be able to gather together. And they're not necessarily coming from top university. But because they are in this network, they can volunteer and do an amazing job. And whenever we ask on Slack, is there anyone that want to help on something, we got people helping out. And so I think this really taught us that we could enlarge our network beyond the traditional group of researcher and a university has been collaborating in the human genetic space for many years, to other group of clinicians, as well as other group of researchers that are traditionally underrepresented in this community, and through this initiative, they can come together and contribute to that. Yeah, so this is all from my side.
Thank you very much indeed. Very useful, very enjoyable talk. I'd like to give the microphone to Carlo Getzi, who had a very interesting question. I'm not sure if I can do this. Yes, answer live, does that work?
Can you hear me?
Yes, we can, excellent.
Oh, okay. No, actually my question was prompted more by the previous presentation. And actually the question is, most of the technology that is being developed to support, the coronavirus crisis, and like the simulation that Nikki was describing before, these systems seem to be based on a number of assumptions that are hidden, that are not explicitly visible, which makes a system different to compare to another one, which makes options hard to combine in a single system to add features into an existing system. So my question is there any effort towards providing guidelines to build these systems in a way that, first, the assumptions are clear, second, that they are composable, so that you can compare the results of your simulations, under one assumption or without a certain assumption, compare what is being done in one country where certain assumptions and certain actions have been taken with respect to others, all things that allow to make these analysis repeatable, shareable. Is there any effort at the EU level to support unification of these systems to be able to handle in a better way the future software that we are developing to deal with the virus?
Andrew, would you like to comment on this?
Well, I think the question was for the speaker before.
Indeed. So if you don't wish to comment, I will ask Nico if he'd like to address this. If Nico is there and can put his microphone on. Maybe Nico is no longer there.
So it was too late. And actually,
okay.
Well, maybe I can just mention there are a lot of initiatives at EU level to harmonize. I'm not sure about the simulation, but certainly EU is pushing to harmonize, for example, these cohort studies across Europe, and I've been trying for many years to do that. And there are some challenges, but I think we will move slowly on that direction.
So I had a very brief question for you, Andrea, and I was very briefly looking at the map with the different data sets you already have. Do you have contacts with China? And if not, would you like me to put you in touch?
Yeah, so China is interesting. No, we kind of were in contact with China through, but they never formalized their participation. And I think the challenge is that China has certain regulation where not only the individual level data of genetics, individual data cannot leave China, but even the summary statistics there are certain challenges in sharing outside China. So that might push them back on that. But if you have content and you think they're interested in participating, that's certainly.
We should discuss this offline.
I have a question, Hannes.
Oh, okay.
If there's no other questions, because I don't want to. If you have other questions, I can wait.
I mean, I'm just wondering, I was trying to read if they're specifically for Andrea. There's some very interesting points being brought up, but if you have a specific question for Andrea, I would prefer to do that.
I think there is a question from Sarah. She's asking, can be challenging to balance privacy with protecting patient health data with promoting international collaboration. Yes, this is certainly the case, and that's been the case in the past. There is a lot of unclarity in the field on what can be and cannot be done. So I think prospective collections, in the collection that has been started to collect now, we have been pushed so that in the end, the patient is the owner of the data, right? So when he reads an informed consent, he needs to be informed on what his data are used for. And so I think in the past, in many of these studies, the consent were very restrictive and very unclear. And so in the new consent that we suggest to do to the different study, we are very clear that if you want to participate, you need to be willing to share your data with the wider scientific community. And so that's how we are navigating that. But there are some old studies that cannot do that simply because they didn't ask that in their consent form.
Thank you very much. Hannes, it's your own timing. Either you can ask the question very briefly or we can move on to the next speaker.
No, there is one question by Oliviero Stock. I have given him the audio. If you...
Excellent.
Oliviero?
Can you hear me?
Yes.
Good, thank you. Thank you for the presentations, very, very interesting. So I think that now in general science and in particular IT has a special opportunity science has got more prestige to the public, and everybody understands that international studies are needed. Of course, science has no borders. This is a given. And so now I would like to get more to politics and science. Okay, health systems belong to states, and states during this crisis have all decided basically to close borders. And I think that this was particularly bad for Europe, because states have prevailed even on Europe. And but of course, the virus recognizes no border. This is also something that people can understand.
Oliviero, could you keep the question short?
Very, very quick then. Nobody is safe, on the other hand, unless the others are safe. So my point is that it would be very nice if we could provide some modeling of closures that is not based on closures along the state borders, but it is along something that crosses state borders. So different borders that cross the state borders would have given comparable, better result, hopefully, than the closures across state borders. I think this would have a great, great significance.
Yeah, no, I mean, it makes sense. I think, like, from a pragmatic perspective, the closure on the border is simply is simple to control, right? Because they are borders, and so you can see who is getting in and getting out. Yeah, I don't see why not use a different clustering approach than countries to reduce the spread. Yeah.
Okay, thank you very much, Andrea.